Hello, everyone. My name is Darla Saunders, and on behalf of the Atlantic Salmon Conservation Foundation, I would like to welcome you to today's webinar. This series is made possible in part thanks to the contributions of the Government of Canada. For today's webinar, we're very pleased to be hosting Dr. Sebastián Pardo. He is originally from Chile, where he grew up fishing for introduced salmonids in local rivers and lakes. He completed his undergrad in marine biology at the University of Queensland in Australia, and then went on to do a PhD on shark and ray life history relationships at Simon Fraser University in BC with Nick Dulvey. He now works as a postdoctoral researcher at Dalhousie University under Jeff Hutchings on modeling Atlantic salmon life histories. After this afternoon's presentation, we'll be opening the floor for a question and answer session. You'll have the option of asking questions directly using your microphone, or you can type them in and we'll read them aloud. I will now turn the webinar over to Sebastián.
Hello, everyone. Thanks for joining for today's webinar, and I'm very excited to be talking about the research I've been conducting over the last couple of years on Atlantic salmon life histories. To begin, I want to say that this research is not just my work, and it's taken the contributions of a lot of people. I'm working under the supervision of Jeff Hutchings, but the data that I've put together and analyzed is being provided by DFO offices around Eastern Canada, as well as the provincial government of Quebec. And there's been colleagues in Norway that have helped with the modeling approach and in the Pacific Coast, and also that my research has been funded through the Atlantic Salmon Research Joint Venture, as well as the Atlantic Salmon Conservation Foundation. So I'm grateful to all these people, and without them, this research wouldn't have happened. So to outline today's webinar, I'm going to provide a brief introduction to life histories, as well as Atlantic salmon and their life histories. And then I will speak about two major aspects of their life histories. First, I'm going to talk about repeat spawning, and then I'm going to talk about marine survival, which is the bulk of the research I'm doing. And then I'm going to briefly mention some potential explanations of the patterns we see, which is mostly just speculation from my part. And I'm keen to hear other people's thoughts on this subject as well. And lastly, I will talk about future directions and where I'm hoping to take this work. So if you look at biodiversity in the world, we see that organisms that have evolved on Earth are extremely different from each other, and the strategies that have evolved to survive and pass on their genetic code and to future offspring is very, very different from each other. And that brings me to the introductory point of what are life histories, because we mention it a lot, but rarely define it. And with life histories, I mean they're age and stage specific patterns and timing of events that make up an organism's life. So I find that we can think about it very easily, for example, when looking at the life cycle of a chicken. It starts as an egg, then the time from when the egg is laid to when it hatches is the time of hatching, which you can measure in days. Then you can measure its growth rate, so how much mass it's accruing over a specific period of time. And eventually this chicken will reach maturity, and you can measure the time it took for this individual to become mature. And then this chicken may lay eggs and reproduce, and you can measure its clutch size or its fecundity, so how many eggs it's laying over a certain period of time. And if we're lucky, this chicken will live to an old age, and we can measure its longevity, so how long is this lifespan. So all these measurable patterns and timings are what we define as life history traits, right? So it's this measurable events and timings are what I'm focused on. And just as a broad introduction to life histories, there are in general three dimensions in life histories. First, they're size related, so the size at which you attain specific milestones as your maximum length, or your length of maturity, or your length at birth. Things like that are size related traits. Then we have reproductive traits, which are to do with fecundity and breeding interval. But the ones that I focus on are time-related traits, which are to do with longevity, age of maturity, mortality, population growth rate. All those are examples of time-related traits. And I focus on these because for some fishes and the ones that have been researched, time-related traits seem to be most closely related to population declines and extinction risk. Right, so now on to Atlantic salmon. I find Atlantic salmon to be really interesting from a life history perspective. We all know that many Atlantic salmon are anadromous, and the eggs are laid in freshwater. They hatch, they spend a bunch of time in freshwater, and then they migrate into the ocean. And in general, the salmon in eastern Canada, if they live for two more years at sea, they will end up going to West Greenland and then come back to the natal streams to hopefully reproduce. In North America, they breed in freshwater rivers from the northeastern US to northern Quebec. I think there's a single population in West Greenland, but that's as far as we know. They tend to spend anywhere between 1 and 5 years in freshwater, and the anadromous fish will spend on average 1 to 3 years at sea. In some populations it's more 1 year, some populations more 2 years. And they spend that time at sea to get bigger, and then they will come back to the rivers to spawn. Many will survive spawning, unlike most Pacific salmon that die. Most of them die after spawning events, then go back to sea, and hopefully if that salmon's lucky, it will come back to the river and spawn again after every year, or they'll take alternate years and take a break in between. So when we look at the life cycle from when they're in freshwater to sea, we can focus on one specific part, which is when it goes from smolt to adult. And this is a stage that I'm particularly interested in, is the survival of smolt salmon, of adult salmon, and particularly as they're measured when they're leaving the river as smolts. And one note about Atlantic salmon that makes them particularly interesting to me from a life history perspective is that they're extremely variable. So they can reach sexual maturity from 7 centimeters long, for example, precocious male par, to around 1 meter in length. This is a salmon that would have spent many years at sea before returning to the natal stream. And concomitantly, the differences in ages of maturity is 1 order of magnitude, from 1 year in a precocious par to 10 plus years, which are some of the salmon in the Tenor River in the border between Norway and Finland. Okay, so now on to the patterns. So what I'm interested about life histories is, we know there's variation in life histories in Atlantic salmon, and I want to know, how do these patterns vary in space and time? So some of the things we already know is that there are patterns in space with regards to river age. So salmon in Nova Scotia, southern populations tend to spend fewer years at sea than in freshwater than salmon in northern populations. And that's a general trend. With regards to sea age, the trend is not as clear. So we know that Newfoundland populations, as well as inner bay and founding populations, for the most part spend 1 winter at sea or 1 year at sea. And then you can have populations in New Brunswick, Nova Scotia, Maine, Quebec that spend 1 or 2 or 3 years at sea. It's more variable there. But then when you think about populations further north, they also sometimes spend longer times. So it's a bit messier, but there are patterns also with regards to how many years they spend at sea. And the interesting thing to me is correlating life history patterns in their life histories with extinction risk. So if you look at this map of the COSABRIC assessments for Atlantic salmon populations, you can see that the populations in northern Newfoundland are green, so they seem to be doing better than the populations in Nova Scotia, Also, the population in southern Newfoundland is doing okay. So you can see that there's also spatial patterns in relation to extinction risk. So perhaps there is a correlation between life histories and vulnerability. So now I'm going to talk about patterns in repeat spawners. We don't know much about this, but fortunately, my colleague Xavier Bordelot spearheaded this work, and he was interested in knowing what were the spatial temporal patterns in the incidence of repeat spawning or heteroparity. And he thought this could be important because, you know, repeat spawners are on average larger fish, which might mean they produce more eggs, and they could have an important contribution to the egg production for a given population. So this work was spearheaded by Xavier, and I find it really interesting, and I collaborate in this, so I'm going to talk about it a little bit. Also in relation to how it compares with what we see in marine survival, which I will talk about later on. So what Xavier did and what we did was we looked at 11 populations of Atlantic salmon in Eastern North America. It's actually 10 populations plus data from West Greenland, which combines, you know, it's an amalgamation of a lot of fish from different populations coming to us in this point in space. So we looked at the return abundances of small and large salmon, and then we basically, looking at a subset of scale data, we assign whether those small and large salmon were maiden spawners or repeat spawners. And then we looked at that for all the years for which we had data for, and we used a discriminant factor analysis to look at trends. And as you can see from the map, the populations are cover a broad area, so all the way from Maine to northern Newfoundland, and even Labrador. So for example, and here is one of the results, is that the proportion of different spawning groups does seem to change a little bit in time. This is data for the Western Arm Brook in Newfoundland. And as you can see, in the 70s, there seemed to be very few repeat spawners. And then as time has gone by, the proportion of repeat spawners for this particular population has apparently gone up. So this is the kind of patterns that are interesting. What are the changes in this proportion of repeat spawners? So focus on the extent of the dark bars that is at the top of each plot. So when we look at the 10 populations, we can see that, you know, they can be quite highly variable. Obviously, for some rivers, the proportion of repeat spawners is relatively low, and much higher than others. But overall, it's interesting to see that for some rivers, the proportion of repeat spawners seems to be going up, while other rivers, it seems to be going down. For example, the La Have River in Nova Scotia, the proportion of repeat spawners seems to have gone way down, while the Western Arm Brook, it's gone up. So we were interested in looking at these trends and comparing them with each other. And also, oh, as a side, I will mention that the data from West Greenland also shows a similar proportion of repeat spawners. So what we did, we looked at the trends and trying to see was there an overarching trend in proportion of repeat spawners, and whether that trend was the same for populations in the northern end of the distribution or the southern. So we found that there was one overarching trend, which is this plot A here, that where the trend is going up in proportion of repeat spawners. And then the loadings, what they show you is whether the trend is positive or negative. So for populations that have a positive loading, it means that the trend is indeed going up, and populations that have a negative loading, actually the trend is inverse and it goes down. And what we found is that this correlated quite neatly with latitude. So northern populations had an increasing proportion of repeat spawners, while southern populations had a decreasing proportion. And this was really interesting, and this work is already published, so the citation is here at the bottom. So I find it, we already know that there are differences in space with regards to repeat spawning, and also we're seeing differences in time with regard with repeat spawning. And these differences in time are not uniform across the distribution of Atlantic salmon. So this is we're just looking at proportion of repeat spawners, but what about trying to translate number of repeat spawners into a metric of reproductive contribution? Because we know that two-winter fish tend to be predominantly female, and also repeat spawners tend to be predominantly female, and repeat spawners are even larger than two-winter fish. So if we try and scale that into thinking about not just numbers of fish, but numbers of eggs, what would that picture look like? So there's only data for 3 rivers to be able to do this comparison, and which are the Con in Newfoundland, the Trinité in Quebec, and the Miramichi in New Brunswick. And we found that for the 3 populations with enough data to assess egg production, that especially in years where there are some years in which the repeat spawners can potentially contribute around half of the total egg production for that year, which is really big. And when we looked at this in a bit more detail, we found that, as you would kind of expect, in years with low maiden returns, you tend to have a higher proportion of repeat spawners. And it seems like the repeat spawners act as a way of buffering reproduction in low maiden spawner years. So its maidens abundance could be quite variable, but the variability of repeat spawners might be much less, and therefore we see more repeat spawners in those low maiden spawner years. So if you keep that in mind, and you know, with what I mentioned earlier, in that the proportion of repeat spawners has been higher, sorry, it's increasing for northern latitudes, that is basically, or I imagine that probably is providing more resilience to those populations, given that, you know, they have that buffering capability in years that for some reason have low maiden spawner returns. Sorry, right. So now to the research that I've been mostly focused on over the last couple of years, which is marine survival. And I'm very interested in this, and particularly by estimating the proportion of fish that survive from small to adult. We know very little about the patterns in marine survival. The literature quite often states that marine survival has declined for most Atlantic salmon populations, but the evidence for it is quite indirect. It often relies on stock recruitment relationships, which are based on making assumptions about fecundity and recruitment, and not very many studies. There are a few, but not very many have looked at in detail patterns within rivers. And for that, you need good data on smolt abundances as well as adult return abundances. So what I did was compile data for as many populations as I could, of many salmon populations as I could in Eastern Canada. And I ended up with 7 time series of smolt abundances as well as adult return abundances. And this range from 19 to 42 years in length. And 2 of them are in Quebec, the Trinité and the Saint-Jean. There's the Nashwack in New Brunswick, the Lahave in Nova Scotia, and 3 rivers in Newfoundland, which are the Western Arm Brook, the Campbellton, and the Con. So the earliest of these time series starts in 1974, but most of them are somewhere in the late 80s, early 90s. So these time series, as I mentioned, are of yearly smolt abundances as well as yearly abundance of adult returns. And like in the example, in the case of repeat spawners, most of this data came in the form of abundance of small and abundance of large salmon. So we then converted to abundance of 1 sea winter and abundance of 2 sea winter fish using a subset of the scales for the years in which the fish return. And what I find particularly interesting is that these 7 populations encompass varied life histories. So the populations in Newfoundland tend to be 1 sea winter dominated, while the populations in the mainland are less so, and also are quite varied in abundance. Some populations seem to be crashing, while some other populations seem to be relatively healthy. So for this, I tried to implement a model that could run for all populations, regardless of their life histories. And this way to try and get at using one approach for estimating marine survival in all the rivers. So I decided to use what is called a Murphy's maturity schedule model. And these are the only 2 equations I'm going to add in the whole presentation. But it's a model that makes a lot of assumptions, but it requires 3 parameters: yearly smolt abundances, yearly 1 sea winter abundances, and yearly 2 sea winter abundances. And basically, if you look at the equations, it's the smolt abundances and time -1 multiplied by the survival the first year at sea times t times the proportion returning that first year in time t is equal to the 1 sea winter fish that returned at that time. And then if you extend that for 2 sea winter fish, that number in time step t plus 1 is equal to the survival of the smolts, sorry, the number of smolts, the survival in the smolts on the year t, times the proportion of salmon that didn't return as 1 sea winter fish, times the survival of the additional second year at sea. So one of the issues with this model is that there's an analytical problem in which you have 3 parameters to estimate, and you're only using 2 pieces of data. So in the past, studies have made assumptions I've used data and separated the sexes. So instead of having these 2 equations, you have these 2 equations for males and these 2 equations for females. However, the rivers for which there is that information are quite limited. So the way I went around it is slightly different. And just to reiterate here, the 3 parameters that we're estimating are the survival in the first year at sea, which is S1, survival in the second year at sea, which is S2, and the proportion returning after the first year at sea, which is called PR. Oh, and also we did not include 3 sea winter fish and older, or repeat spawners for this. So it's just looking at the abundances of 1 and 2 sea winter fish. So to get around that analytical problem, we use the basin implementation of it, which basically allows for the use of prior knowledge to be incorporated. And this is really important because these priors and this prior knowledge can have an impact on your resulting parameter estimates. So just to show your priors for the parameters S1 and 2, these are our priors. So we're basically saying that we believe that the survival in the first year at sea is less than the survival in the second year at sea. And likewise, the proportion returning, we know that for a 1 sea winter dominated river, the proportion returning is going to be much higher in the first year than for a river where most fish are 2 sea winter or older. So we use different priors and we incorporated them in this model, where for populations that are 1 sea winter dominated, we expect the proportion returning to be relatively high. And for other rivers, we made the prior relatively wide. So we say it could be anywhere around here. So because of this prior curves, the model can converge, and it will, based on this priors, you'll get parameter estimates. Right, so now to the results. So the first thing I want all you to notice is that these trends are quite variable. So for some rivers, it seems that the current estimates of S1, that is marine survival in the first year at sea, seem to be relatively high compared to previous years, while other rivers, especially the Con, the Lahave, and the Nashwack, seem to have much lower S1 estimates now than they did before. Some rivers, the Saint John, they're relatively high in comparison to in the early 90s. And it's, this is not necessarily new, because these estimates of marine survival for the once sea winter dominated rivers are really similar to the return rates. So we already knew that, the return rates in the Con are declining and the return rates of the Western Arm Brook are not. One thing that's important to point out is that we don't have data for this period before the 90s, except for the Western Arm Brook. So we really don't know what was going on there. I think a lot of the literature that talks about marine survival and declining compares it with what was happening in the 70s. But at least with this data, we can't really assess how current levels of marine survival in the first winter are compared with those historical values. But the picture in the last 30 years seems to be that, some rivers are going up, some rivers are going down, some rivers are all over the shop. And if we were to overimpose all these trends on top of each other, we'd see that things are quite messy, and you really can't say that, at least for these 7 rivers, you're seeing much of a pattern in regards to marine survival. Again, we can't really compare with what was happening in the 80s and 70s. And also, these lines, the vertical lines, are the years of the moratoria, the commercial salmon fishing moratoria for these provinces. And there's, you can't see anything that pops up to mind. Now, when going to survival in the second winter at sea, the picture is very different. So for 7 rivers, the estimates are really uncertain, particularly the once sea winter dominated rivers, Con, the Campbellton, and Western Arm Brook. I should probably just ignore those estimates for those rivers, given that the proportion of 2 sea winter fish that are returning are extremely low. So you would expect that a model that is generalizing across all life history strategies will be really bad at estimating survival in the second winter at sea for populations that have very few of this fish. So as you would expect, the model has very uncertain parameters, which are, if you look at the distribution, is really similar to the prior for this parameter. So the data is not informing the estimates for S2 for the Campbellton, the Con, and the Western Arm Brook. To some extent, the Nashwack and the Lahave. But the patterns in the Saint John and Trinité pop up. These are rivers that seem to have more of those fish. And still, I now don't feel very comfortable talking about survival in the second winter at sea because of the uncertainty associated with these estimates. So for the most of what I'm really interested in is the pattern in survival in the first winter at sea. And now when we look at the estimates of proportion returning, well, the model seems to be pretty effective at showing that, those rivers where it's mostly once sea winter dominated, the estimates are 1. Again, informed by a very tightly bound prior, but we still doesn't deviate much from those estimates. In the Saint John, we estimated the proportion returning is pretty low, while in the Lahave, the Trinité, and the Nashwack, it seems to be quite variable. And interestingly, in the Nashwack, it varied quite a bit in this year. So I don't know if this is an artifact of the data, but something funky is going on. And also in the Lahave, the last few years seem to have a lower proportion estimates than the previous time series. Again, I should mention that there are gaps in this data because every now and then you have years where you weren't able to estimate smolt abundances because the water flow was too high, so your counting fence got washed out. So you have no estimates for the year. So we basically excluded those years from our data set because we'd have to make some very big assumptions otherwise. And one of the things I did for the model, especially with regards to the proportion returning that I should have mentioned earlier, is that it was a hierarchical model in that we estimate population levels estimates of returning rates. And again, as you can see, for the 170 dominated rivers, the estimates of the proportion return are really high, very low for the Saint Jean, and the other rivers are somewhere in between. And again, the variability in terms of their coefficient of variation is really high for the Nashwack, a bit high for the LaHaye, and really low for the other ones. So what I was really interested in looking at was the correlation between temporal trends of every combination of river. I wanted to see if marine survival is declining uniformly across rivers or increasingly uniform across rivers, and if so, which rivers. So I did a very simple Pearson correlation test to assess this, looking at the median values of those trends. And for the years where we have overlapping years, these are the values from the Pearson correlation coefficient. And if you look at this first graph, the one about significance, you can see that the color ones are the only significant correlations, which are positive, but overall, most rivers are not correlated. And if you look at directionality, which is the second plot, you can see that they're mostly blue, but it's really a mix of red and blue. So some rivers are positively correlated, some are negatively correlated, and only 3 of them are significant. I want to mention the caveat, though, that the Pearson's correlation coefficient is on median values of marine survival in the freshwater North Sea. And this might not be the best way to look at correlation. So I'm thinking about other ways of doing so better because we might still be underestimating it. And other models who've looked at marine survival in different ways have created matrices that allow you to estimate this correlation within the model. But in the case where we don't have overlapping time series and we have missing years, that is much more mathematically complicated to do. So we have to estimate this correlation after the fact and not within the model. Ideally, I would like to have that parameter within the model, but given the type of data, it's not that straightforward. Right. So what does this mean? And the first thing that comes to mind is, at least for the 7 populations that we looked at, the trend in marine survival in the first winter at sea, that is S1, seemed to vary considerably within and among populations. So I was expecting to see declines everywhere, but this does not seem to be the case. Could be that those rivers aren't representative of what's going on over the whole population, but at least those rivers, and I'm just focusing on the 7 rivers, we don't see this uniform trends. If there is an overall decline in marine survival since the 90s, it is not uniformly occurring for all populations. So this is basically the same point. We really can't say much about what was happening before. It could be that that marine survival now is much lower than it was back in the 70s, but from this modeling approach, we can't tell. And what are the potential explanations of this? I mean, it could be that the declines happened in the late 80s, early 90s, and now they're at a low level, so we aren't able to detect them because of the length of our time series. So that's one explanation. Could also be that our estimates of marine survival are just plainly wrong because of the assumptions that we made and the methodology we use. But our estimates of S1 are almost the same as the Newfoundland return rate, so it tells you the model is effective at extracting that information. And also the estimates of S1 and S2 for the Trinité are really similar to the 2-sex maturity schedule model that Gerald and colleagues implemented in their 2003 paper. So this kind of tells me that, at least I'm doing it. I'm extracting estimates that are coherent and similar to what other people have estimated for other rivers. The other thing is, if there is a region-wide decline in marine survival, so if overall the pattern of survival across all salmon in eastern Canada is going down, we're seeing large within and among population variability. So the variability in marine survival is relatively high with regards to any trend that might be occurring. So this is really interesting because it tells you that the patterns are not uniform, they're highly variable, and if there is a region-wide declines, you're not going to see them necessarily on a river-by-river basis. And the other explanation that I think seems reasonable, given that we don't see any correlation between closely related populations, is that marine survival in the first winter at sea is probably strongly influenced by local conditions. And as I mentioned in my introduction, this is just speculation. We really can't say what the case is, but I'm just thinking out loud here and looking at potential explanations of why this might be the case. And lastly, this results made me think about what about the correlation between climate and population trends? Because we see there's a lot of work that has correlated climate variables to abundance trends, not survival trends, abundance trends. So what could be the mechanism there? And I really don't have an answer for that because we're not necessarily, we don't really know what's going on. And my hunch, and this is another speculation, is that there are perhaps things going in the ocean that don't just impact survival of individuals. There are perhaps non-lethal effects that cascade further into the life cycle of the animal, such as, food provisioning and the effects towards potential reproduction in the future. Again, all speculation, but I think this results prompt me to think about what else could be going on. So with that, I'll just leave some concluding thoughts about Atlantic salmon life histories, and that their life histories are changing in Eastern Canada. As you can see, the proportion of repeat spawners in rivers in Atlantic Canada is changing differently for rivers up north and rivers down south. But yeah, life histories are not a stationary thing. And and likewise, marine survival is constantly varying for these rivers, and it's doing so differently for the rivers that we examine. Some of marine survival has been going up, some of them have been going down, and some of them are all over the place. The other thought is that marine survival might not be declining uniformly for all populations in the region, at least not since the early 90s. And it also prompts you to think, okay, there's this big idea that marine survival is the reason why salmon, one of the main reasons, or one of the important reasons why salmon have been declining, and it probably is. But what are the other factors that are impacting populations? And especially, what interested to me, what are the other factors of oceanic conditions that are impacting populations? Because it's very likely that ocean conditions are not just being manifested in population dynamics of Atlantic salmon via just survival, and there could be some other non-lethal effects. And the work in progress for me, and what I'm going to be working in the next few months, and I'm already working on, are correlates of marine survival. So what things are correlated with the trends that we see? Temperature is the big one that comes to mind, and I'm going to just show you a little bit of this, which I've already been working on. Life histories is another one. It could be that there's evidence that if the smolts are coming out of the rivers at a larger size, they tend to have a higher survival. Also, I was just thinking, how do trends in marine survival correlate with trends in repeat spawning. Perhaps the evolutionary circumstances or the fitness landscape, to put it one way, is acting similarly on those two parameters. So those are correlations that I want to further explore. And also, I'm very interested in density-dependent process. How does marine survival relate with the overall abundance of salmon in a given river? And when looking at temperature correlating with marine survival, I'll just show you some of the limited exploration I've done, which is kind of to be continued. Is this graph shows you the monthly sea surface temperature for May, June, July, and August for 6 rivers, and the y-axis shows you the estimates of S1, so marine survival in the first winter at sea, and for that given year. So we're correlating for every estimate of marine survival. We're looking at what was the temperature on the ocean just by the river mouth for that given year. And we don't seem to see very much of a pattern except for the Nashwack River, which is in the highest end of the spectrum when it comes to temperature, where you can see a negative relationship between marine survival and temperature. But for the other rivers, you don't seem to see this, which was kind of baffling. But at the same time, you know, I wasn't really expecting to see much here because these are two noisy parameters. Marine survival is a noisy parameter, and SST is noisy in how it's expected to impact marine survival. So these are the kind of correlations that I'm interested in exploring. And if you have any suggestions of potential correlates that I should also be looking at, I'd be more than happy to hear about them. And with that, thank you very much for your time, and I'd be happy to answer any questions.
Thank you so much. That was a great presentation. Lots of food for thought. So the question and answer period is now open. So you've got 2 options for asking your question. You can use the webinar control panel, which is the little gray box you should be seeing in the upper right-hand corner of your screen. If the box is minimized, just hit the orange arrow and that will enlarge it. So if you're using the audio of your computer, you can figuratively raise your hand, which is the hand icon, and we can unmute you so that you can ask your question directly. Or you can type in your question on the control panel and we can read it aloud for you. So I can see we've already got a few questions here in the queue. The first one comes from Frank Johnson, who asks, are the Inner Bay of Fundy salmon a simple case? They stay in the bay.
Yeah, I mean, they do stay in the bay, and but that doesn't necessarily make it simpler for estimating marine survival, because it's all about the counts. So how many smolts are coming out, how many adults are coming back in. So I'm not sure what you mean with simple case, in that, yeah, they just stay in the bay or the Gulf of Maine, but for estimating marine survival, it might not be that straightforward necessarily.
Thank you. The next question comes from Guillaume Dauphin, who asks, or says, hi Seb, thanks for the update on your work. Regarding your input data, any sort of insight discussion points on how smolt estimates are generated? Are methodologies from each of the rivers you looked at the same, or are there some differences? This could potentially make the survival signal more noisy. Thanks.
Yeah, so I didn't go into detail about that part of the model, but I am accounting for observation error in smolt estimates. And yes, the way smolt data is gathered for rivers is different. Some use, absolute counts, some use marked recapture methods. There is observation error, which accounts for the uncertainty in those estimates. So for rivers where we have a smolt abundance estimate with uncertainty, we use that uncertainty in the model, which is included. So that usually ranges in the 5 to 8% variation, and that's included in the model. So we're accounting for that uncertainty. For rivers where we don't have that information, we're assuming a blanket 5% observation error uncertainty. Yeah, I should have probably mentioned that, but I figured that aside. But yes, I have explicitly incorporated that uncertainty in the model. And there's also uncertainty in returns estimates that I've incorporated as basically coefficient of variation.
Thank you. We now have a question from Xavier Bordelot. Hello, you can ask your question now.
Yes. Hi, Seb. It was nice. Hey, nice to see your talk. Thanks. I was just thinking back at the plot you showed with the comparison of the time series for survival for the first year at sea. And then I was thinking about if you ever tried to standardize these time series, because I don't remember what the peak looked like. I know your signal, you were saying that it wasn't fitting to one and another in terms of the different population, but I'm just wondering if you tried standardizing them like we did for trend and repeat spawning, and if there was a better fit using that.
I did that, and I looked at and I didn't mention that the correlation matrix that I showed you is on standardized. So
oh, okay, I see.
So they are standardized. It probably won't make much difference when it comes to calculating Pearson's correlations, because that will standardize them anyways. But yes, I have done that, and it doesn't look, and if you were to plot them, it doesn't look very different, and it just looks all over the place.
Oh, okay. Yeah, thanks.
Thank you.
Our next question comes from Robert Battis Jr., who asks, what about freshwater flow effects on salmon survival for affecting local conditions?
I think that's really important. I do think that if we had that data, that'd be wonderful to include in correlates of marine survival, because I expect for years with very little freshwater flow, you might have some effect on marine survival, but being able to get at from this modeling perspective, I'm not sure if we have that information. Also, when smolts are being counted, in general, I imagine they're already relatively far downstream, so a lot of the mortality could have already happened. We can't really tell. But yeah, it's definitely important from survival in the freshwater part of their life cycle, and especially the out-migration. But given the different ways in which the fences are set and the data that gets collected differently in each river, it's hard to be able to generalize, say, for this river, there will be a strong correlation with freshwater flow, and for other rivers where the smolt are counted right at the river mouth, there might not be, because mortality might have already been happening by the time smolt are counted.
Thank you. Our next question comes from Peter Solonius. Peter, would you like to ask your question?
Hello. I wanted to make a comment about the Nashwauk, where there's in most of the years there's a pretty good monitoring of smolts going downstream and at least grilse returning. In the early 2000s, when the salmon farmers were treating sea lice with emamectin benzoate or slice, we started to have a spectacular increase in ocean returns. Then towards the end of the 2009, 2010, the sea lice became resistant to that chemical, the sea louse disinfectant, and our returns from the ocean crashed again. So we see some circumstance that suggests that sea lice are very important as far as smolt survival is concerned. Would you like?
What was the last bit? I missed the last bit of your.
We see quite a bit of a concern with sea lice abundance when the salmon farmers cannot control any longer the sea lice numbers as they did in the early 2000s. We see a crash again in ocean returns. We had spectacular increases in the mid to late 2000s.
Yeah, so that it is really interesting you mentioned that, because I wasn't aware that there was a correlation in the Nashwauk. Those estimates of marine survival for that period are very high, the highest out of any river, any time series, any time period that I analyzed.
Yes, one of the years I think we had a 12% return
Yeah, which is crazy.
It is absolutely crazy. But as soon as the salmon farmers lost control of sea lice, we were back to dismal returns again.
Thank you for sharing that. And that speaks to the idea that, you know, there is very, very high variability and in a local scale with regards to marine survival. So, you know, the conditions in some rivers might be good one year, bad in the next, and good again. And while others that are perhaps not that far away, the trend could be totally different. And this is what I think is really important to at least think about with regards to the data presented.
Thank you. Our next question comes from Alex Bilak. Alex, are you there?
Yes, I am. Hi there. Thanks very much for a very interesting seminar. I'd like to, first of all, I think, congratulate you on being cautious with interpretation of data over what is a fairly decent time span, but in the work that I did when I was a grad student in my day with Jeff Power, doesn't necessarily tell the tale. And we actually examined 120 years worth of data from the Godbout and a slightly shorter data set from the adjacent St. Marguerite, as well as a bunch of other Quebec North Shore Atlantic salmon rivers. And you could only really discern what was happening over a far longer period than some of the shorter records. Now, I think that you're probably on the cusp of having some good stuff, but when I look at my own graphs here, I see that, you know, you could end up with some false conclusions. So good on you for being cautious with that. The second thing I'd like to say, and this perhaps might be of interest to Mr. Bordelot as well, is that I donated the entire scale collection of samples from my work on the Quebec North Shore, which are held, I believe, in the New Brunswick Museum. And those are available, I think, for anyone to look at or research. If they could get something useful out of those, then they're there for people to use.
Thank you very much for that. I'm very curious about the data that you collected. Do you have, you know, when you say you went back that really long time series, what specifically were you looking at return counts, or was there small
differences between age classes of fish, and knowing, for example, that in most Quebec North Shore salmon rivers, which were controlled by angling clubs, that grilse didn't count. They were equivalent, treated as the equivalent of trout. It was a garbage fish, and they really weren't recorded in most fishing records, with the exception of one camp on the Godbout, the Molson camp, and that was spotty as well. So I was able to demonstrate a very significant change in proportions of 2C winter versus previously spawned salmon, and also demonstrate a decrease of about a pound in mean weight over the period of between 1865 and 1985 for 2C winter and previously spawned salmon. I did look at relationship between river age and sea age and all that sort of jazz. It's all in my thesis, obviously. Some of the historical records were published in the literature. And I also dabbled a little bit in life history strategies in terms of looking at the influence of river harshness, that paradigm that's suggested by Paul Elson and Bill Schaffer. And their basic thesis was big long rivers equals big fish. And I began to look at it from a slightly different perspective, looking at how difficult the ascent of a river might be and how that might influence size or morphology or what have you. It was quite striking that there were 2 or 3 rivers on the Quebec North Shore that were either renowned for very large salmon or renowned for a branch of a specific river having very big salmon. And the classic of that was the east and west branches of the Moisey River, where the guides could easily distinguish an East Branch fish from a West Branch fish just by looking at it. As a biologist working on the river, I could see no difference until I was sat at a camp for probably a period of 2 or 3 weeks, looking at all of the fish that were captured. And finally, one day, I was sort of just thunderstruck that it was so obvious. I took a whole bunch of morphological measurements, but ended up using just length and weight correlation. That was not published.
I would love to hear more about this. If you could send me a quick email just so we can
you can get a hold of me, alex@blac.ca.
Okay.
And you can find some of my stuff in CJ/FAS and other journals.
Yeah, it'd be really good, interesting for me. Now I'm thinking also, how could I be able to compare this trends in this shorter time span that I've been able to explore? Because really we're constrained by accurate small counts. And, you know, back in the day, there's a lot of information of adults, but very little information on smolts. So yeah, it'd be really interesting to compare these trends with, you know, what was perhaps happening in the past. So thank you for sharing that.
Thank you. Our next question comes from Paul Stowe, who asks, did you analyze smolt length and SST, or average smolt length and marine survival?
I have looked at smolt length and marine survival, and my preliminary analysis has shown no correlation. I think it's mostly because mean smolt length is quite noisy, so it doesn't really vary consistently from year to year, but it's something that I will definitely go back into now that I have more data on more rivers, and also thinking about other ways of looking at smolt length and not just you know, mean size. I think we're losing quite a bit of information by averaging, so I'm also interested in the variance, or perhaps are the smolts, you know, do the smolts cover a broader range of sizes, just looking at meal size? But yeah, my preliminary analysis, there wasn't any obvious relationship, but I do want to dig deeper.
Thank you. The next question comes from Stanley Oliver, who says, hi, I noticed the farthest northeast you have studied is rivers in Newfoundland. Are there any plans to incorporate the Labrador salmon rivers?
I would love to get data from Labrador. I don't know if there are any future plans of collecting smolt abundances for the Labrador rivers. If there were, I would be delighted to do that. And I think perhaps we should be focusing more on Labrador because, you know, as we know, a lot of fish are moving their distributions north, given, you know, they're tracking the change in climate. And it seems that Labrador will probably, if not already, is going to be the base for the healthiest Atlantic salmon populations out there. So I think we need to, I would love to, you know, be able to look at, you know, potential ways of counting smolt and counting adults and setting up that so we have a better idea of what marine survival is for those populations. I don't think the data is there yet, but I think we should aim for it.
Great, thank you. The next question comes from Shannon Murphy. That was a great presentation demonstrating a regional analysis. Do you plan to do any correlations with genetics for your repeat spawning analysis? Also, do you think supplementation stocking programs could be confounding the genetics with respect to the repeat spawning analysis?
I think, I don't know how we would incorporate genetics for the repeat spawning analysis. Can't think off the top of my head if there's enough data across a time series to do so. I mean, it would require a lot of sequencing. But with regard to your second question, I completely agree that there could be some genetic introgression that's impacting you know, the composition of repeat spawners for a population, and we wouldn't be able to tell. And I think the same thing is also happening with marine survival. I imagine that for rivers where there are a relatively high proportion of escapees that are making their way into the gene pool, that you'd expect the fitness of those smolts, especially at sea, to be much lower.
Okay, and a follow-up question. Do you think supplementation stocking programs could be changing the proportions that you are observing?
Yes, But I think we aim to balance that, and we did not include stocked salmon in, at least not in the marine survival comparisons, and I'm pretty sure we didn't in the repeat spawner analyses either. We removed those stocked fish from the analysis wherever we could.
Thank you. Okay, so I've got a couple of follow-up comments to previous questions. So Stanley Oliver has written that he would love to chat sometime about your interest in incorporating Labrador salmon rivers.
I would love that. Yeah, I think that's where we would need to be focusing and seeing what's going on up there, given that salmon are moving, or the distribution is shifting north. So my email is right on the screen. Please feel free to send me a quick email, say, hey, this is me, and then we can correspond.
Thank you. And we have, I think our final question is a follow-up question from Robert Vadas Jr., following up on his question about freshwater flow effects on salmon survival and local conditions. And so he's written, freshwater flow does affect salinity in coastal habitats. What are your thoughts about that?
Oh, that's interesting. Yeah, I think the effect of salinity could be quite complex in that there's probably going to affect what kind of predators are there, what food availability there is for the salmon. If it's less saline, maybe they have less food. Off the top of my head, I don't know how you would expect changes in salinity is resulting from freshwater flow to affect survival of migrating smolts. I would love to hear what you think about it, because I can't think off the top of my head of how that would pan out. But sure would be really important. If we have the data, I would love to look at if there's any correlations. The main question, the main issue with looking at correlations is getting the data, or the year-by-year data for each river to test the correlation for. And I think that is like the constraining issue.
Great. Okay, thank you. Thank you again for a terrific presentation and for a very lively question and answer session. So I